Nathan Price. I am the Associate Director and Professor at the Institute for Systems Biology in Seattle. My main expertise is in computational systems biology. So what excites me most is that the threads of systems biology, which have been developing over the course of time, have really come together to where I think we can really deliver important solutions for people and get them translated out into the world. There's been an enormous increase in the ability to generate data, especially that is relevant to human health, which is the area that I work in the most. And the ability to generate deep phenotyping, or what we call personal dense dynamic data clouds around individuals, pulling together information from genomes, from their proteomes, metabolomes, microbiomes, clinical labs, wearable devices, and being able to take a very holistic view gives us new approaches to thinking about how we can understand the underpinnings of the earliest warning signs for disease, so we can get in there and try to predict them, try to prevent them. And so that's the ultimate goal of much of what we're doing, which is to try to reverse disease in its earliest stages before it actually becomes a problem. What's really going to drive needs in our field is to take that complexity and be able to apply algorithms and pull out actionable possibilities. What do you actually do from this, so that we can deliver with simplicity insights to individuals about what was really important for health, if we're in the health space, or for engineering microbes, if we're trying to do something in the energy space, or whatever it is. But can we take that complexity, analyze it, understand it, conceptualize it, and then deliver something that's actionable to a person? There isn't such a thing as an ideal data set. There's always issues. So the measurement has its own impact on the system. So there are these limits, right? No matter how you do a measurement, you are perturbing that system. And that gets to be difficult in terms of reproducibility. There are other issues because every biological system is in fact unique. So when you go back into the same biological system, it's not really the same. It's either the system at a different point in time, or it's a similar system that's genetically a little bit different. Right? Every human is, of course, very unique in terms of both genetics and history. So all of those things make analyzing biological data complex. And so there is no such thing as an ideal data set. I do think, but there is obviously the idea of data sets that are much better. So there's a lot of effort right now going into, for example, the analysis of electronic medical records. But there are pretty severe issues with it because it's not actually designed to be a transfer of information the way that we think about computationally. And this is why we've really been focused on generating more uniform, large, what we call personal dense dynamic data clouds, so basically deep phenotyping, lots of multiomics and dynamic and wearable device measurements and so forth that are fairly uniform. Because our idea is if you can structure your data in this way, get really a dynamic, big set of data like that, that basically generating more regularized data of the kind that you really want to see, you might be able to learn a lot more by having those kind of data structured to learn from than just focusing on the past of electronic medical records or something like this that's not actually structured to have what you want to have. So I do think that there's no ideal data set, but you want to think about the data that will catalyze the future that you're trying to create, and you can certainly have improvements and have better data sets. But what COBRA does is it brings mechanistic modeling at scale, and that's really an important thing. So you can build a genome scale model, and you can represent all the metabolism of an organism, and you can't do that in a lot of other approaches. And it sort of fits in between, say, your really detailed mechanistic, kinetic type models, which are very hard to build for systems above a certain size. And it's also contrasted with your big statistical type approaches, which you can do at arbitrarily large sizes, but don't have that same grounding and mechanism. And so it gives you a nice structure to think about what do we know from the standpoint of mechanism in a cell and then utilize that as a means to understand the data that you're analyzing. So it can get into a lot of areas that people care about. So if you fuse, for example, the kind of things that we have in constraint-based modeling with machine learning or artificial intelligence types ideas, this can get you to understandable or explainable AI, or XAI is a very hot topic. Well, this actually has some relevance to that. It gives you structured big data analysis, which is another area that everybody cares about in the data analysis and biology field. It gets you at the integration of multiple omics types, because you can hang all of those on this network framework. And that's really important, because otherwise each individual omic is scattered in different databases. We have databases for transcriptomics and for proteomics, and there are practical reasons for that. But networks force you to come together. And the other thing that's really important about constraint-based modeling, or about this field, is that it is a structure for trying to understand how far you are in understanding processes, either in a cell or a body or whatever it is, and do you have enough information to explain what you're seeing or not. And often if you have data that's scattered across biology papers, you don't actually know that in the 7th paper you read actually is in conflict with the 12th paper, or that those things don't fit together in a network context. And you don't know that until you start building a network and making predictions. So the framework for modeling is actually extremely powerful for thinking about the future of biology and where it should go. So one thing I would say, maybe I'll aim this towards students or people who are coming into this space, is that it is an extraordinarily exciting time to be in science. The world in biosciences in particular is changing very rapidly. The ability to make big impact is perhaps never been better. And so I would really encourage people, if they want to have a life in science, that it's a time to get involved.